The platypus. This one won't kill. That's why it's first. It weighs about as much as a hardcover book. It lives in Australian rivers. It looks genuinely like someone assembled it from spare parts. And it produces a venom that modern medicine cannot treat. Not because the venom is deadly, but because of a single molecular detail buried inside it that breaks the treatment process entirely. The venom is delivered through spurs on the hind legs of males. It contains a mix of proteins, including a C-type natriuretic peptide, a signaling molecule that triggers sustained breakdown of mast cells, meaning the cells that regulate pain and inflammation. The result is immediate, severe, and spreading pain that does not respond to opioids. Morphine doesn't touch it. Victims have required regional nerve blocks, the kind used during surgery, just to manage what a river mammal the size of a shoe produced. The venom also contains something stranger, a D-amino acid. Amino acids are the building blocks of proteins, and they come in two mirror image forms, L and D. Almost all biology on Earth uses L-amino acids exclusively. The D form is the structural opposite, built backwards at the molecular level. It functions, but the immune system doesn't recognize it as something to build antibodies against. There is no antivenom for platypus venom because one of its key components is a molecule. The immune system, as currently understood, cannot learn to neutralize. That hyperalgesia, the extreme pain sensitivity, can last for months after a single envenomation. There is one more detail. Only male platypuses carry venom, and venom production peaks specifically during mating season. This is not a hunting tool, not a defense mechanism against predators. The biology produced a compound that defeats the strongest painkiller in modern medicine for the purpose of male competition during breeding. That D-amino acid is a structural accident that happens to make treatment impossible. The next animal on this list didn't arrive at the same problem by accident. Its venom defeats antibody production by being too complex, not too strange. The antivenom process has one core requirement. The venom must be a protein the immune system can identify and target. Every animal from this point forward found a different way to make that impossible. African bush viper. Hundreds of antivenoms exist for venomous snakes. The global effort to address snakebite has produced treatments for cobras, mambas, vipers, rattlesnakes, and dozens of species most people have never heard of. The African bush viper, genus Atheris, has no antivenom. Not because it's obscure, because the genus is too variable for any single antibody library to cover. The venom is hemotoxic and cytotoxic simultaneously. Hemotoxic means it attacks the blood. Cytotoxic means it destroys cells directly. What it targets specifically is the clotting cascade, the chain of proteins the body activates to seal damaged blood vessels. The venom consumes those proteins faster than the body can replace them. This is called consumption coagulopathy, a failure of the clotting system, and it means the blood can no longer stop itself from flowing. Internal bleeding accelerates. The kidneys, filtering blood that the circulatory system can no longer manage, begin to fail. In 2021, a wildlife care specialist at the San Diego Zoo was bitten by an Atheris. The treatment team had access to every antivenom available. They tried several. None of them halted the progression. The specialist survived on supportive care alone, meaning manage the symptoms, protect the organs, and wait. The confirmed death count from African bush vipers is unknown. Not uncertain, unknown. Most bites happen in remote Central and West African rainforest, where medical documentation either doesn't exist or doesn't reach the literature. The absence of data is itself a measurement of how little the medical system has been able to engage with this animal. The Atheris genus is diverse enough that the venom composition varies significantly between species, and to some degree between individuals. Any antibody developed against one sample may be irrelevant against the next. The bush viper's untreatable status is not a resource problem. It is a structural one. The next animal doesn't have variable venom. Each individual carries a completely unique chemical library. The problem isn't diversity across the population, it's complexity within each animal. A beach, warm water, a shell on the sand, marbled, symmetrical, beautiful. The kind of thing people pick up and photograph. This one will kill before the photographer reaches the shore. Geography cone snail. It is called the cigarette snail. The name comes from researchers and herpetologists, people who study these animals professionally. And the implication is that after a sting, a victim has roughly enough time to smoke one cigarette before dying. In practice, severe envenomation takes 1 to 5 hours. The nickname survived anyway, because nothing else quite captures it. The geography cone, Conus geographus, is a marine snail found in shallow tropical waters across the Indo-Pacific. It looks like a collectible shell. It hunts fish. It stings with a harpoon-like structure called a radula, which can reach any direction from the shell's opening, including backward. People handling them from what they believe is a safe end have been stung. Each cone snail carries up to 200 distinct peptide toxins, short protein chains, called conotoxins. The exact mixture is unique to each individual animal. Alpha conotoxins disable the receptors responsible for muscle contraction. Other components attack sodium channels, calcium channels, serotonin pathways, and adrenergic, meaning adrenaline-related reward systems simultaneously. One researcher described the combined neuromuscular effect as equivalent to being bitten by a cobra while consuming pufferfish. Two separate and incompatible poisoning mechanisms firing at once through a formula that is different in every animal that carries it. Because the cocktail varies between every individual snail, any antivenom developed against one sample is partially irrelevant against the next. The target moves every time. By 2016, researchers had counted 36 confirmed human deaths, all attributed to C. geographus. Mortality in untreated severe envenomation approaches certainty. The LD50 of conotoxin, the dose lethal to half of a test population, sits in a range that fits invisibly on a pinhead. One conotoxin, isolated from a related species called Conus magus, has been FDA approved as a painkiller under the brand name Prialt. It is among the most powerful pain medications in clinical use. The pharmaceutical industry extracted the chemistry, synthesized it safely, and brought it to market. The snail that carries it in a cocktail with 199 other compounds remains completely untreatable. The cone snail's venom is not one molecule. It is a library that rewrites itself in every individual. The next animal has a single toxin, one molecule, and that simplicity is precisely the reason nothing can be done about it. Everything so far has involved venoms that are too complex, too variable, or too structurally unusual to neutralize. The second half of this list is different. These animals produce venoms that aren't just hard to counter. They are structurally immune to the anti-venom process itself. Blue-ringed octopus, the mechanism behind why the hospital cannot help. The answer is not about how potent the toxin is. It is about what the toxin is made of. Tetrodotoxin, TNTX, is not produced by the octopus. It is synthesized by symbiotic bacteria living in the animal's salivary glands. The octopus is a delivery system for a bacterial weapon it did not build. TTX is 1,000 times more toxic than cyanide. A single adult, blue-ringed octopus carries enough to kill the equivalent of a mid-sized office building's worth of people, with nothing available anywhere on Earth to reverse it. TTX blocks voltage-gated sodium channels across the nervous system. Sodium channels are what nerve cells use to fire. When they are blocked, signals stop reaching muscles. The diaphragm, the muscle that drives breathing, stops contracting. The victim remains fully conscious, aware, thinking, unable to inhale. The progression continues until the brain is deprived of oxygen long enough to lose function. The bite is often painless. There is frequently no visible mark. Antivenom works by training the immune system to produce antibodies against a protein target. TTX is not a protein. It is a small organic molecule, small enough that the immune system does not register it as a foreign body worth attacking. There is nothing for the antibody process to attach to. No antivenom exists because the molecule is structurally invisible to the mechanism that would create one. In 2006, a 4-year-old in Australia was bitten twice by a blue-ringed octopus. His mother identified the animal and reached a hospital within 20 minutes. Staff intubated him immediately, put a tube down his airway, and used a machine to breathe for him. 15 hours later, he regained voluntary muscle control. That sequence of events, identification, speed, and mechanical ventilation sustained for 15 hours, is the complete treatment protocol. There is no other option. TTX is invisible to the immune system, so no antivenom is possible. The next animal found a more disorienting problem. Its venom is visible to medicine. The immune system can detect what it does. and treating it the standard way can kill the patient faster. In the water, warm, clear, no sensation. 25 minutes later on the shore, the spine begins to ache. The skin starts to crawl, the chest tightens. There was no moment of being stung, there is no mark. Something has already happened. Irukandji jellyfish. It is 2.5 centimeters wide, transparent. It drifts through tropical water without being seen. The initial sting registers as almost nothing, a faint prickling at most. The syndrome it causes, named after an Aboriginal people of far north Queensland, is something emergency physicians describe as one of the most distressing presentations in acute medicine. The delay is part of what makes it dangerous. Symptoms arrive 20 to 30 minutes after envenomation, by which time the victim is often out of the water, on shore, away from any obvious cause. The venom activates presynaptic sodium channels, the channels nerve cells use before a signal fires, triggering a massive, uncontrolled release of catecholamines, stress hormones, specifically norepinephrine and epinephrine, from the body's own nerve endings. The venom does not damage the body directly. It commandeers the body's emergency response system and locks it at maximum output. Blood pressure surges to dangerous levels. Heart rate spikes and stays spiked. Severe pain radiates through the chest, abdomen, and back. The presentation is clinically indistinguishable from a major cardiac event. In approximately 2% of cases, congestive heart failure develops within 12 to 24 hours. The heart fails not from direct toxicity, but from being forced to operate at crisis intensity for too long. No antivenom exists. But that is not the most dangerous part of the clinical picture. When a patient arrives at an emergency department appearing to have a cardiac event or severe allergic reaction, standard protocol may include epinephrine, adrenaline. In Irukandji syndrome, administering additional epinephrine into a system already flooded with catecholamines can precipitate end organ failure. The reflex that saves lives in other contexts becomes a risk here. The antivenom problem is not molecular in this case. The mechanism of the venom makes conventional emergency treatment a potential liability. The syndrome was confirmed through a method no institutional review board would approve today. Dr. Jack Barnes, to prove that a specific jellyfish species caused the condition, placed the animal on his own forearm, his 9-year-old son's forearm, and a local surf lifesaver's forearm. All three were admitted to hospital. 2 confirmed deaths in Australia are attributed to Irukandji syndrome. The real number is almost certainly higher. The syndrome produces symptoms that match heart attack. Without a known exposure history, it is routinely misattributed at autopsy. The Irukandji recruits the body's own emergency chemistry as the weapon. The last animal on this list does not need to recruit anything. It attacks the heart directly, and it has had an antivenom for 50 years that may not work. There is an antivenom. It exists. It has been stocked in Australian hospitals since the 1970s. Researchers are not certain it does anything at all. Box jellyfish Chironex fleckeri, the most venomous animal on Earth by any measure of lethality per encounter. It has no brain, no nervous system capable of intent, no target. It is a mesh of water and protein and tentacle, drifting, and contact with a large adult specimen can stop a human heart within 2 to 5 minutes. A single adult trails up to 60 tentacles, each extending up to 3 meters. Each tentacle is lined with millions of nematocysts, spring-loaded venom delivery cells, that fire on contact with any surface, regardless of what that surface is or whether the animal has registered it. The venom proteins punch pores through the membranes of heart muscle cells. These pores allow potassium to leak out of cells faster than the body can compensate. The result is hyperkalemia, excess potassium in the bloodstream, which disrupts the electrical signaling the heart relies on to maintain rhythm. Ventricular fibrillation or cardiac arrest follows. Many victims do not reach shore. At least 70 confirmed deaths have occurred in Australian waters, partly because antivenom access in remote coastal areas is limited. The antivenom was developed in the 1970s from immunized sheep. It is real. It is in hospitals. Independent clinical reviews have been unable to confirm that it meaningfully changes patient outcomes. The reason appears to be this: the venom proteins denature, break down, rapidly when extracted from living tissue. The molecule that enters the sheep to generate antibodies may not be structurally identical to the molecule the jellyfish actually delivers during a sting. The antibodies are trained against a version of the toxin that may not exist in the field. The real molecule arrives without anything in the patient's system capable of stopping it. Medicine built a cure for a molecule that only exists inside a living animal. In 2019, a CRISPR-based genome widescreen identified a zinc ionophore, a compound that blocks the pore formation mechanism that prevented venom damage in mice when administered within 15 minutes of exposure. It is not in human trials. It exists as a research compound. The most venomous animal on Earth remains in clinical practice without a confirmed working treatment. The anti-venom process is over a century old. It has saved millions of lives. It works because most venoms are proteins the immune system can be trained to destroy. What these animals represent is not a gap in funding or effort. It is the outer boundary of what that process can reach.